What I would like to do is introduce you to viruses in general and the coronavirus that we're all worried about in particular. So I'm going to be doing this with reference to my own lab's work on HIV-1 because our research into coronaviruses is more recent. Okay, so first I wanted to get some nomenclature down. COVID-19 is actually the disease caused by the current coronavirus, which is called SARS-CoV-2. So you can see in this view, this is a schematic of the virus, but this is actually an electron micrograph, so this is more accurate. And the virus, the coronavirus family of viruses in general, were named for the fact that they have these spikes that stick out. A lot of viruses have those, so that's nothing new. But the coronavirus spikes are really large and they create this corona-like effect or a crown. So it was named after that. Now I want to say some caveats. The first is, in my personal opinion, I would recommend that you get information about COVID-19 and this particular virus from scientific websites or from the Center for Disease Control or the World Health Organization, or preferably from your doctor. I don't recommend news sites. I've seen some of the science is incorrect on several news sites I've looked at. And I also want to say I'm not an MD, so I'm going to concentrate on the science, which I'm trying very hard to get correct. Okay, so first I wanted to say that of course the world has experienced pandemics over the years, as you're well aware. And I wanted to highlight from this nice graphic here, what they're doing is showing you in proportion the death toll for these various pandemics from highest to lowest. And so what you can see is that the bubonic plague was the worst that the world has experienced, at least in recorded history, but there were others that were quite bad as well. And I wanted to highlight smallpox. I'm going to be saying something about possibly reusing the smallpox virus as a vaccine vector for COVID-19 disease. Also, as you've all heard about, the 1918 flu killed a lot of people around the world. And then is still ongoing. HIV that causes AIDS has killed probably up to 35 million people since that pandemic started in the 1980s. And then here we are with COVID-19. This graphic, unfortunately, is out of date, so more people have died than this. It might not look like much, but unfortunately, I'm afraid that death toll is going to get higher. And then MERS and SARS are caused by coronaviruses that are related to SARS-CoV-2, the virus that's causing COVID-19. This is Middle East Respiratory Syndrome and Severe Acute Respiratory Syndrome. These have pretty much, they're not happening anymore in the world. At least MERS isn't, and SARS maybe a few cases. So I wanted to mention these because on the next slide, you'll see that SARS, MERS, and SARS-CoV-2 are related to each other. They descended from bats, and that's common for coronaviruses in general. So the initial host of these viruses were bats, which harbor many different coronaviruses, and these sometimes pass into humans. In particular, they often pass through intermediary hosts, and so the intermediary host that passed into humans was probably a civet or a raccoon dog for SARS. For MERS, it was camels, and for SARS-CoV-2, it might be a pangolin, but people aren't really sure right now. But eventually these change enough so that they're able to infect human cells. I want to point out here that these viruses here, which are also coronaviruses, cause common colds. So these four right here, these got into humans maybe hundreds of years ago, possibly even 1,000 years ago, and now they've attenuated to the point that they cause a lot of the colds that children get and adults get as well. So about the biology of the virus, coronavirus is an enveloped virus, and that means that it has a lipid membrane around it, which is shown right here in this picture. It's this red part. And so this means that the virus itself acquired that membrane from the host cell. So that's something that the virus picked up from the host cell. It also has genetic material, which in the case, viruses can include either DNA or RNA. where DNA is the code and RNA is the message that allows you to actually make proteins. So this is an RNA virus. And then I want to point out that the spike protein, which gives it its coronavirus name, it's a very large protein. So that being said, this is all in common with HIV, HIV-1 that is, and many other viruses. HIV-1 happens to also be an envelope virus. It's an RNA virus, and it has spikes on its surface. There are a lot of differences between the two, and HIV-1 is not a coronavirus. But what I want to say is that the purpose of any virus, including SARS-CoV-2, is to get into a cell, make copies of itself, and then get out and spread to other hosts. And so a virus is really a parasite. It doesn't include enough of its own machinery to be self-sufficient. So you can't describe a virus as being alive, whereas a bacterium, for example, is self-sufficient and it can grow on its own. A virus needs a host cell. And in the case of SARS-CoV-2, it has adapted so that it can use human cells as its host cell. So that's the purpose of a virus. Get in, hijack the cell machinery, make more copies of yourself, and then transmit to other new hosts so that they can make more copies. So it's a hijacker. And of course, that's what HIV does. It gets in, makes more copies of itself, and then gets out. But I want to point out that one of the things my lab studies for HIV, and that we'll be studying for SARS-CoV-2 as well, is how it gets in. And in the case of HIV, it's been known for decades now that what happens is that the spike on its surface, which is a trimer, it's depicted over here. The SARS-CoV-2 spike is also a trimer, meaning it has 3 identical subunits. Its receptor is a host protein called CD4. That makes the HIV-1 tropism limited to cells that express CD4, and these are cells in your immune system. So when HIV infects you, you wipe out the cells in your immune system, and then your immune system doesn't really work anymore, and you're susceptible to all kinds of opportunistic infections, which leads to AIDS, acquired immunodeficiency syndrome. Okay, so for the coronavirus life cycle, the receptor is not CD4. It was discovered incredibly quickly by researchers that since this came out in January or so, the world became more aware of it. Its receptor is a protein called ACE2, which is on the surface of, for example, lung cells, and it's in other places in your body as well. So what has been discovered also in remarkably short time is that the spike protein, when it interacts with ACE2, in order to actually enter the cell, it needs to be cleaved by this protease, which is called, a protease is an enzyme that cuts other proteins. That's what these scissors mean. That protease is called TMPRSS. Now notice this paper from David Wiesler's group at University of Washington. This is from 2017, so it couldn't have been done for SARS-CoV-2. It was actually something that was known for SARS and some common cold coronaviruses, but it turns out a number of these use the ACE2 receptor. So let's talk about ACE2. So as I said before, ACE2 is used as a receptor for SARS-CoV-2 to enter human cells, but SARS also uses it, and so does at least one of the common cold coronaviruses. So ACE2 means angiotensin converting enzyme 2. What that means is that it cleaves angiotensin. And that's a peptide hormone that controls vasoconstriction and blood pressure. So more about that later. But let me just say that it's known from previous research that ACE2 is on the membranes of cells that are in the lungs, the arteries, the heart, the kidney, and the intestines. So you may have been reading that there are often GI problems in COVID-19 disease. There can be heart problems, kidney problems, and so on. And where the receptor is expressed, what types of cells, that can determine, what goes wrong in this COVID-19 disease. Okay, so the fact is that ACE2 is important in controlling blood pressure, and so the interaction has been people have been worried about perhaps taking ACE inhibitors if you have high blood hypertension. And I just wanted to say that the American Heart Association has said unequivocally, do not stop taking ACE inhibitors if you are currently taking them. The reason I'm mentioning this is that they may have effects such that they lower or raise the levels of the ACE2 receptor. So they affect the levels of the ACE2 receptor on the surface of their target cells. And that could contribute to lung damage. So people were somewhat worried about taking these inhibitors. And the doctors are telling you, do not stop. And please talk to your doctor about this if you're considering it. On the right, what you're seeing is something from a structure of it's a three-dimensional structure. This is what people do to visualize interactions at the atomic level between two different proteins, let's say. And this is actually a structure that was solved 15 years ago now for another coronavirus. It was actually the SARS coronavirus, and this is the receptor binding domain of SARS actually in this cyan color. There's a receptor binding motif here, and then this is the structure of ACE2, and that was solved by a group at Harvard 15 years ago. So now this is available for SARS-CoV-2. It actually looks very similar. So people are trying to figure out differences between those at an atomic level right now. Okay, so back to the HIV life cycle. What we're going to talk about now is something general for all viruses. They have to make copies of themselves. So that's step 2. So I wanted to point out that there are enzymes that do this, and the enzyme that does it for HIV-1 is something called reverse transcriptase. This makes copies of the HIV viral genome. Now, a little aside here, HIV is an RNA virus, but it makes copies into DNA, which is the message, and then it goes in and it does something really terrible because it's what's called a retrovirus. It puts the DNA copy into the nucleus of the cell. It actually integrates it into the chromosome. And what that means is the cell can, not even be producing virus, but it's got a copy of the viral genome. So at any time in an HIV infection, the cell can start making new viruses. So you cannot get rid of an HIV infection with antiviral drugs because it is integrated in its genetic material is integrated into your own cells. And this is what's really horrible about this type of virus, which is called a retrovirus. And as many of you may know, the discovery of reverse transcriptase was done by David Baltimore when it was a long time ago, I believe. I forget what year, but David was 37 years old when he got the Nobel Prize for this discovery. The reason it was really an amazing discovery was that at the time, the central dogma of biology was DNA makes RNA makes protein. So the idea was the genetic flow of information would only go from DNA into RNA, and then RNA would give you the information to make the protein, and the protein would do the business. This was reversing that flow of information, RNA going back to make DNA, and he discovered this for a different retrovirus than HIV, but it turns out to be what HIV also does. Okay, back to the point about reverse transcriptase. It makes a huge number of errors. This results in an enormous number of viral strains, even within a single infected individual. And this is a problem, a horrible problem for HIV, because that means that it's very hard to make a vaccine. It's very hard to make antibodies or protective immune responses against so many different strains inside you. Now, fortunately for the coronaviruses, they actually don't make that many errors. And the point I made here was that the HIV reverse transcriptase, which we would call an RNA-dependent DNA polymerase, has no proofreading activity. So things that copy DNA, like in our own bodies, it's really important for them to not make mistakes, or these would result in mutations. Mutations are not good for a single individual usually. So we have proofreading activities that fix those mistakes. this virus has no proofreading activity. In fact, as I'm going to tell you, coronaviruses do, and they are unique in RNA viruses in having that sort of proofreading activity. And if you think about it, it's in HIVs. HIV, if I can anthropomorphize here, wants to make that many mistakes. It wants to make a lot of different viruses, and then it is survival of the fittest. It makes all kinds of viruses. Most of them don't work well, but the ones that do go on to make new strains, and then your immune system is all confused. So back to the coronavirus now. The coronavirus only copies its RNA message into RNA. So it doesn't have a DNA form. So it is not a retrovirus. So it has what's called an RNA-dependent RNA polymerase. And that's associated with a proofreading enzyme, which that's unique among RNA viruses to my knowledge. Okay, so that's going to be important for thinking about possible drugs that you could use to combat coronaviruses. So speaking about drugs, I want to illustrate that although it's hard to make antiviral drugs, there are very successful antiviral drugs for HIV. And so those have been developed over the years. There are many different inhibitors of reverse transcriptase, the enzyme that copies RNA into DNA. These are just analogs of the building blocks of the nucleic acid. So these are what are called nucleoside analogs. These are building blocks related to building blocks of the DNA. So many of these exist. And then there are inhibitors of integrating the RNA into the nucleus, into the DNA. There are then protease inhibitors. I'm going to come back to this later because this is the thought that you could inhibit a viral protease is being considered for SARS-CoV-2. So often viruses require their own protease to cut up their own proteins. And this is an absolute requirement for HIV. It cannot survive unless it has its protease, so there are inhibitors against the HIV protease. And then there are inhibitors that stop fusion from taking place, which then means the virus can't enter. Now, the important point about the HIV drugs is you must give at least 3 at a time because HIV mutates so fast that it can, if you give it just one drug, it will mutate to become resistant to it. So if you give it 3 or 4 drugs, it can't necessarily mutate fast enough. And as you may know, in this antiretroviral therapy for HIV/AIDS was available to people starting in 1996, and it was pioneered by David Ho, who's a Caltech alum and I think still a trustee at Caltech. He was Time Magazine's Man of the Year for this. I think that was in 1996. Okay, so you have to give 3 at once because of the very high mutation rate of HIV. I think it might not be a bad idea to give several at once for the coronavirus. It also mutates. It doesn't mutate as much because of this proofreading activity of its enzyme. Okay, so now I want to talk about antiviral drugs that are in development now that might help control the COVID-19 disease. And the great thing is that some of these are already FDA approved for other illnesses. And so these they've been tested in humans. They know about its efficacy and safety for these other diseases that they're being used for. And so these could be ready, and some of them are even in people now. So that's a great thing, is that if we can take already approved drugs and use them, there's a hope of really quickly going after this disease. Okay, so I wanted to mention several places in the coronavirus life cycle, and in particular the SARS-CoV-2 life cycle, where the drugs are being tested. So the first of these is an inhibitor. Remember I said there was a protease called TMPRSS. This is actually at the surface of the cell, and it is a host protease. The host makes this. But it turns out that actually this spike protein has to be cleaved before it becomes active. And so if you inhibit TMPRSS, it appears, at least in vitro, that this virus can't enter as well. So that's one thing that's already approved drug. So that could be used perhaps in treating COVID-19 disease. Now there's another protease that's in an intracellular organelle called the Golgi apparatus. This is where proteins pass through during their biosynthesis. That enzyme is called furin. And there's an interesting thing about SARS-CoV-2 spike compared to the SARS spike, and it appears to be cleaved by furin to activate it on its way out. And so people are trying to develop antifurin reagents. Now, I'm sure you've all heard of chloroquine and hydroxychloroquine. I want to discuss another point of the SARS-CoV-2 life cycle, which involves going into these compartments inside the cell that are called endosomes. And these are actually acidic. So I'm pointing them out here. And in this very busy slide that shows you the life cycle of coronaviruses in general, this was, of course, from a 2019 article published in the spring, so they didn't know about SARS-CoV-2 at the time. But they were pointing out that coronaviruses go into this low pH compartment called an endosome. So it's important the pH is low. So at least from the research I've read, SARS-CoV-2 probably also enters endosomes. At least I've seen some evidence that says it does in vitro experiments. And this might be relevant to why weak bases, such as chloroquine and hydrochloroquine, these are currently licensed for use as antimalarial drugs, and also, as I'm going to say in a moment, for some autoimmune diseases. So these are already licensed, so this could be an off-target or an off-label use of it for SARS-CoV-2 infection. Okay, so if you raise the intracellular pH of this, some of the enzymes that are critical in here probably don't work, but that might not be the whole story, or it might not even be any of the story about if chloroquine and hydroxychloroquine actually are efficacious. They might be doing this by just in general reducing inflammation. These drugs are being used in patients with autoimmune disease, such as rheumatoid arthritis or lupus. And so there's some thought that the pathology related to COVID-19 disease, when people get what's called acute respiratory disease syndrome, or ARDS, is that your immune system in general, does all these things when you're infected with a virus, which are trying to get rid of the virus. And one of them is to give you a fever, to produce more mucus, to make you cough and sneeze. That's the idea, is to make you expel whatever infected you. Sometimes it gives you, if it's a gastrointestinal pathogen, it will give you diarrhea or make you vomit to get rid of the agent. So there are all these things that happen that the pathogen actually doesn't do. It's your own immune system, but it's there to protect you. Well, unfortunately, it appears to go quite off when people have a very severe reaction to SARS-CoV-2 infection. As you've heard, in about 20% of the cases, it leads to this acute respiratory disease syndrome, and the people who are dying have a horrible reaction from their own immune system. And so the idea is that you could possibly reduce inflammation, and that's why they're also trying to use an existing drug against one of these, it's called a cytokine, it's produced by your immune system, and it creates inflammatory events. It's called IL-6, interleukin-6. And so a company has made an antibody against IL-6 that's used for autoimmune diseases. That's being tested for COVID-19 treatment. And from what I've heard, it might be more efficacious if you first put the person who's in severe distress on on some prednisone or another steroid that also would reduce inflammation. And so that's all being tested. I don't know if the story's out on that. And all of these have to be done by real clinical trials. But anecdotally, I think I've heard interviews with doctors in New York that say this may be a promising treatment. Now, another potential drug target is the viral protease itself, this is not a host protease. So not to confuse this with, I was talking about TMPRSS and also furin. These are in the host cell. They're there to cleave their own proteins, but they also cleave the SARS-CoV-2 spike. But there's a viral protease as well. And just like in the case of HIV-1 infection, you can, one of the drugs that's used to treat HIV-1 infected people is a protease inhibitor. This was developed a long time ago now, in the 90s. There are a series of these. But now some of these HIV protease inhibitors, for example, leponavir, darunavir, these are being tested for whether or not they might work to inhibit the SARS-CoV-2 protease. So this requires a lot more testing. You can actually try and see if you can predict what protease inhibitors that are already approved might work if you have actually the real structure of the viral protease, and then you can do drug design to try and see if you could fit in known inhibitors and so on, and then test these in the lab and then later in clinical trials. Okay, now there's another thing that your body is trying to do to prevent infection by this virus. So as soon as you get infected by a virus, you go through first what's called an innate immune response, where you don't recognize the particular virus itself, but you see that there's something weird going on in cells because there's a lot of viral nucleic acid that shouldn't be there. And that triggers what's called the innate immune response. And it's called innate because we share a lot of the components of this with invertebrates, such as, flies or worms or so on. This is distinct from what we call the adaptive immune response, which is specific for a particular pathogen. And now I'm talking about antibodies, and I'm also talking about a type of lymphocyte called a T lymphocyte. I'm not going to really talk about T cells here. I'm going to concentrate on antibodies. So if your body can make antibodies, it takes a while. It takes 5 to 7 to 10 days for people to do what's called seroconverting, which means that detectable in their blood, there are antibodies to SARS-CoV-2. And during that 5 to 10 days, you need to have some kind of immune protection, so at least you have the innate immune reaction. Now, the adaptive immune response is specific to vertebrates. So that means, birds, mammals, us, mice, and so on. Okay, so we can all make we can make antibodies. Some of these are what are called neutralizing antibodies. And that means that they block the interaction between the viral spike protein and its receptor. And if you block that, the virus can't get in. So none of the rest of these things happen, which would be great. So let me tell you a little bit more about antibodies. These are proteins in your blood. They combat pathogens. So this is something that, as I said, is part of the adaptive immune response. And these antibodies, through an amazing recombination at the DNA level, allow you to make vast numbers of different types of antibodies. So if there's a new pathogen that has never been seen before on Earth, you can make an antibody against it through this amazing way that we have, that vertebrates, that is, specifically, let's talk about us, humans, or the mouse model system. We can make at least 10 to the 16th different types of antibodies. We don't make all of them at once, but if we need to, we can make antibodies against anything. Okay, so that's being depicted here by different colors of antibodies. So as you may have heard, there are 2 ways people are talking about using antibodies to combat COVID-19, and the general principle is called passive immunotherapy. This means that rather than wait for your own immune response to create an effective neutralizing antibody response, which might take too long to help someone, you give them antibodies from someone else who has recovered from the disease. And so this passive immunotherapy has been done for, for a long time. Actually, if you have a rabies virus, if you're bitten by something that's rabid and you have to have a rabies treatment, part of it is passive immunotherapy. Okay, so this is not a new idea, but how it would work for COVID-19 disease is that you could perhaps get, well, and this has been done in some tests, you get convalescent serum from people who've recovered from COVID-19. And this contains what I'll call a polyclonal mixture of antibodies against SARS-CoV-2. And the reason it's polyclonal is when you make an antibody against a virus or anything else, you make lots of different antibodies against it. You don't make just one kind. So you make this, it's called a polyclonal response. So that's illustrated here by lots of different types of antibodies. Okay, the other thing you can do is you can take the serum or plasma from a patient and you can actually isolate defined antibodies. And if you isolate a defined antibody, that's called a monoclonal antibody as opposed to a polyclonal antibody. So let's say this person had made all these different antibodies here, and a researcher got the plasma from that person. They can isolate cells that would make all these different antibodies, do single cell sequencing of those cells, they're called B cells or B lymphocytes, get the genetic sequence of a particular antibody, and then in the lab that can be produced as a recombinant protein. And so this is how, in some cases, antibodies are identified in particular viral diseases. So you might decide, you might isolate all these different antibodies here and you test them in vitro and you decide that maybe this pink one is the best one, but you could also produce, you know, the purple one or the brown one, whatever, and you could give it as a mixture. But this is a defined way of doing this. There are advantages and disadvantages of both of these. This can be done right now from convalescent serum. Problem is you can't drain the person completely and there aren't enough, you know, there just isn't enough convalescent serum to do this. In the long run, it might be better to do individual antibodies or mixtures of individual antibodies, but that's going to take a while. Now, we've been collaborating with Michelle Nussenswag's lab at Rockefeller University for at least 10 years to actually do this for anti-HIV antibodies. They do single cell B cell cloning to isolate particular antibodies against, for example, we've been doing this for HIV-1 and also Zika. And we also have an ongoing effort to look at these antibodies against hepatitis C virus and so on. Okay, but we're engaged in a collaboration now with the Nussenswag lab for them to isolate antibodies against SARS-CoV-2, and then we would look at them bound to the SARS-CoV-2 S protein structurally. And so let me just show you what that would look like in the case of HIV-1. So this is an example of something we're doing for HIV-1. This is a structure, and these are, we have resolved this to the level of individual atoms. And we're looking at two different antibodies. It doesn't matter their names, but their names are over here. And these are bound to the HIV spike protein. And what you're seeing now is the distribution of carbohydrates on that protein because it tries to hide most of its surface with carbohydrates. Those carbohydrates are put on by the host machinery. So I'll just run that again. And so it's trying to hide its protein surface so that antibodies can't react against it. That is a very common trick of viruses. SARS-CoV-2 and SARS and so on do that as well. Anyway, so once we know where the antibodies bind, we can understand how they bind, which ones are good, which ones are not so good, and so on. Now, the other thing we can do is we can note that if we want to make a vaccine by, for example, let's say injecting this particular HIV spike protein, there are parts of it that produce what we call distracting epitopes. These are parts of the protein that are exposed, but they're not conserved from one strain to the other. And of course, antibodies target these, and then these are not useful because they only work against certain strains. So I'm saying this because this gives you a context then of what we're trying to do for HIV-1, and now we're trying to do it for SARS-CoV-2. So by having these structures, we can either engineer them using protein design to make them more efficacious. And we're also engineering immunogens that you could actually inject into eventually, maybe as a vaccine, to elicit neutralizing antibodies. These have to be so-called broadly neutralizing antibodies in the case of HIV-1 because there's so many different strains. And here I want to point out that these sorts of structural studies are only possible at Caltech through support of facilities. For example, the Caltech Molecular Observatory, which is a facility that allows researchers at Caltech to solve structures by X-ray crystallography, and the Caltech Cryo-EM Center, which allows researchers at Caltech to solve structures by a method called cryo-electron microscopy. So without the support of these kinds of basic research facilities at Caltech, my research and the research of other structural biologists at Caltech would not be possible. Okay, so let's talk a little bit more about neutralizing antibodies. I showed you that what happens for SARS-CoV-2, it also happens for HIV. If you have a neutralizing antibody, it blocks entry and then nothing else happens. So this is great. People raise these when they're infected with HIV. But in the case of HIV, most neutralizing antibodies neutralize only a subset of HIV-1 strains. And this isn't going to work because in a single infected person, there are more strains of HIV-1 than there are influenza strains in the entire world. So this isn't going to work. People raise these antibodies, it's not going to work. But we're hoping that if we engineered them, perhaps we could get them to be so-called broadly neutralizing antibodies. So for HIV-1, we need them to be broadly neutralizing antibodies. So this leads us to the question about, well, as you've probably read, SARS-CoV-2 also mutates. So can it make mutations that would confer resistance to antibodies? And yes, it can, but compared with influenza, which, as you know, is a virus that comes in a lot of different strains, SARS-CoV-2 exhibits relatively few mutations. So let me just say first that we have influenza vaccines, but they have to be changed every year. So researchers go around, they identify the 3 or 4 strains that they think are going to be most prevalent in the world, and they make a vaccine in a variety of ways against it, and that's why you have to keep repeating your influenza vaccines every year. Okay, so flu has a relatively high mutation frequency, but it's nowhere near as high as HIV-1. Now look at SARS-CoV-2. Now what you need to look at here is not the slope of these lines because the y-axis is different in all cases. What you need to look at is these bottom numbers here. How many mistakes or mutations are made on average per 10,000 units, which in the case of this is a unit of the genetic message or the nucleic acid. Okay, so flu's up at 35 per 10,000. SARS and measles are actually quite similar. So let me tell you something about measles. This is this anecdote, which is really instructive. So there's something called immunological memory, and this is the basis of why there are certain types of viral diseases you can get only once. Chickenpox is a good example. Measles is another. There's something called immunological memory. So when you make an antibody response, in some cases, you make these what are called memory B cells, and they hang around for a long time. And then if you get re-exposed, you can immediately make a immune response, or you can more quickly make an immune response. So if it would normally take you 5 to 7 days, you might have died by that time. But if you have memory B cells, you would do it a lot faster and it would confer protection. So here's an anecdote that shows you how that can work in the best possible case. So in 1781, these sailors came to the Faroe Islands and they were infected with measles, and the people from the Faroe Islands got sick and a lot of them died. And then in 1846, then they had no contact with measles between 1781 and 1846. At that time, another ship comes in, more measles infected sailors, but people who were old enough to have gotten the measles and survived from the 1781 infection they didn't get measles because they had lifelong immunity to measles virus. Now, this is incredible. We make a really great response to measles. We don't have immunological memory for some other viruses that last as long. So, for example, from what's known for the common cold coronaviruses, probably the immunological memory is shorter. It might only be a year, but it's still, there is some immunological memory, and that's what gives us hope for making a vaccine. It might have to be, you'd have to figure out how often to administer it, but I think there's hope for making a coronavirus vaccine. It just may not work as well as the measles vaccine, which will give you lifelong protection. Okay, so what about a vaccine? So I've sort of alluded to this, but most current vaccines work by inducing antibodies against a virus. So you inject something into what's called a naive individual, that is, they haven't been exposed to that virus, or maybe it's a bacterium. And then the immune response makes antibodies to it, and some of these go into these memory B cells, and then if you're exposed to it later, you're protected more quickly. So that's how it's thought that most vaccines work, and it's actually more complicated than that. As I've done more reading, like, I have to say, the immune response to viruses is extremely complicated. We're just learning more and more about it all the time. There needs to be a lot more basic research to understand how our natural antibody response and our natural immune response works for viruses. But in the case of some vaccines, it's believed that antibodies are the so-called correlates of protection. So the question is, you give a vaccine or you get infected yourself, and if you're protected against a re-exposure to that, what is protecting you? And so it's thought that antibodies are often a correlate of protection. So what about a vaccine for SARS-CoV-2? So let's go back and think about vaccines in general. In the US and sort of Western countries, we think about vaccines as invented by Edward Jenner. So he noticed that milkmaids who were exposed to cowpox from milking cows didn't seem to get smallpox, which, as I showed you in the beginning, one of the beginning slides, was an enormous problem hundreds of years ago. So he had the idea that, well, let's just inject people with cowpox. That would be an example of what's called a live virus. It can replicate, but it's attenuated, so it gives you less of a disease or maybe no disease at all. Okay, so that seemed to work. So that's attributed as the first vaccine. Just as an aside, he later experimented on other children, including his son, and these sorts of experiments are obviously illegal today. But actually, immunizations against smallpox were introduced over 1,000 years ago through a process called variolation. In cultures, in places like China, they would introduce dried smallpox scabs into the nose of someone who hadn't been infected, and that person would get a milder form of the disease, but then would be immune to smallpox. It wasn't perfect, so 1% to 2% died after variolation, but that was compared to 30% who died from smallpox. Okay, so what about a vaccine for SARS-CoV-2? Well, we don't have one yet, but I think it's kind of promising that researchers were able to make a vaccine against MERS coronavirus. Remember, this is the one that it's not infecting people anywhere right now. The intermediary host, it went from bats into camels. And this was a vaccine against, for camels, that is. So they made this vaccine, and this picture from the journal article shows that they were assessing how much mucus was formed by the camels. I didn't actually know they developed mucus in their nose, but apparently they do. So they claimed that this was successful in the camels. People aren't trying to develop MERS or SARS vaccines these days because these viruses are not a problem for the world health these days. But what this was it's worth looking at this. It was actually something related to, they used an attenuated virus, meaning it's not going to make the camels sick, but it was actually the vector for it. So that's a term that we use for how we might insert MERS proteins. It was a derivative of the smallpox vaccine. So the virus is actually called vaccinia. It's used, meaning it's used for vaccines. And they just put some genes for MERS into the vaccinia virus. They changed it so it couldn't replicate anymore. And that was used as a so-called vector for vaccine, for injecting into these camels. So this pox vector or vaccinia virus can be used for non-pox related diseases, which I'll come back to when I talk about potential SARS-CoV-2 vaccines. Okay, so that leads me to believe that there's some hope for making a vaccine, at least in this way. So I want to share with you a PDF that I found from the WHO. It's a draft landscape of the potential candidate vaccines, and this was accurate as of the 21st of March. I'll bet there's even more now. They had a disclaimer on their website. They don't necessarily advocate any of these, and they're just telling you what's out there. So I wanted to put their disclaimer here on this slide. So there's two, at least as of today, I believe. It could change tomorrow. But there are at least two that are in clinical evaluation. That means in people. And so one of them was made in China by the Beijing Institute of Biotechnology and a company called CanSino. It's in phase 1 clinical trials for just safety right now. And it's based on using a vector from adenovirus. So it's not a pox vector, it's a different type of virus that's sometimes used for these types of vectors for vaccines. A previous thing that was very similar was tried for Ebola. And then you may have heard about a US company called Moderna. They're collaborating with the National Institutes of Health. This is National Institutes of Allergy and Infectious Disease, which is what Tony Fauci heads up. So, so they're collaborating with the NIH, and they've made what is called an mRNA-based vaccine. It's put in lipid nanoparticles. That's what LNP means. It just means they put the genetic message encoding, they don't tell you this here, but it encodes the SARS-CoV-2 spike protein. That's what's necessary to get into a host cell. And so the idea is the message would go into your own cells would make the proteins, all kinds of immune reactions would happen, and hopefully you'd do all kinds of stuff, including making neutralizing antibodies that would prevent interaction of the SARS-CoV-2 spike protein with its ACE2 receptor. This company has done this for multiple other things. I believe they're trying this for influenza virus. This is a very fast way of doing it because it's very easy for them to make messenger RNAs. They have to alter them a little bit so they can't be cleaved in vivo, but normally they're very sensitive to getting cut up and stuff. But it's pretty fast, I guess, to produce sufficient quantities of messenger RNA. It's quite hard to produce very large quantities of the proteins that the messenger RNA encodes if you wanted to inject that. So that's why they were able to do this so quickly. I'll just go through very quickly. There are other companies that are trying to do this with DNA instead of RNA. And there are, as of when this PDF was out, 48 candidate vaccines in preclinical evaluation. So not in people yet. This means in animal models. And sometimes it means just in vitro at the experimental bench in cells. Okay, so there's DNA-based vaccines. I'm only giving you some of these 48. And then there's an inactivated virus. So one of the flu vaccines is just inactivated flu viruses, like you kill them so they can't infect you. There's another live attenuated virus vaccine. That's the idea of using cowpox instead of smallpox. Then there's subunit vaccines, and that means you produce, for example, the S protein, the SARS coronavirus, the SARS-CoV-2 S protein, and you inject that into a person. And like I said, that's being tried for HIV and also for influenza. Actually, there's a licensed influenza vaccine, which is a so-called subunit vaccine. But like I said, it takes a while to produce large amounts of protein that are what's called under good manufacturing protocol, GMP type of production. So it doesn't have other things in it, which could be very dangerous for you. Okay, and then there are other types of viral vectors where you put the SARS-CoV-2 into the backbone of a different type of virus. And I'm giving you examples here that I picked out, and some of these have been tried for other viruses, and you can see there who's doing them, and you should just keep your eyes out. This is a pox vector, like I was talking about. Okay, so I want to close by saying a few things. So one of them is, this has been an amazing time, even though it's so awful for the world and for people who are getting sick and infected. I have to say that it's been an interesting and in some ways good time for science, because I've noticed a lot more collaboration with a lot of people in many different fields are now coming together to try and work on this pandemic. So I want to thank them. And I'd also like to thank people in my lab who've now switched to SARS-CoV-2 projects, which are allowed to proceed at Caltech in a limited capacity if they're related to COVID-19. So this is a subset of my lab that are working on this. And I really want to thank them for working on it, and to thank the rest of my lab who are attending, giving us ideas, attending lab meetings virtually. I wish I had time to talk about all of them and what they're doing. I'd also like to say, I'm sorry if I didn't put all the references in. I wasn't able to cite all the possible work that people are doing in both around the world and in my lab. And so I'm sorry I'm not going to name all the people, but I wanted to thank my lab for doing science remotely, thank the COVID-19 researchers in my lab. And this is a recent picture. This is people in my lab and also people in the Protein Expression Center, another Caltech facility that is this is the only reason why we can make all these different proteins for vaccine design and antibody research. And this is used by multiple labs across the campus. Okay, and then I wanted to point out Christopher Barnes, who's heading up the COVID-19 research in my lab, and also Anthony West. They fielded questions about this and are helping me with answering a lot of these questions. But I wanted to give you some references for some of the things I talked about. There are multiple other references. These are references in case you're interested in the candidate vaccines, and also a compilation of drug trials and vaccines. Okay, so thank you very much for your attention.